Hi, everyone. So in Chapter 9, Part 1, I give you a bird's eye overview of the process of cellular respiration. And so in this video, this Chapter 9, Part 2 video, I'd like to go into each part of cellular respiration to give you a little bit more in-depth analysis of what's happening. Now, if you're watching this in the fall of 2020, you do have a quiz that's on Friday, September the 18th, and it will cover all of chapters 8 and 9. So please don't forget that that is happening this Friday, September 18th. Now, as a review from chapter 9, part 1 video, we can divide cellular respiration into 4 basic parts. Okay? The first one is glycolysis, and I'm going to abbreviate. The second one is, goodness gracious, is pyruvate oxidation. I'm going to abbreviate here, okay? The third part is the citric acid cycle. And then the final part is the electron transport chain, okay? Remember that this here is happening in the cytosol. Once this part happens, then the products are transported into the mitochondria where the rest of the process occurs. Okay, so let's look at what's going on from the beginning all the way to the end. And the plan here is to follow a single molecule of glucose as it goes through this process and is eventually turned into ATP. So let's start out with the glucose molecule, and you'll recall that glucose is a 6-carbon sugar. So I'm drawing the 6-carbon sugar for you here. Now in the body, this would be in a ring form, but for purposes of ease to understand what's happening, I'm going to start out with 6 carbons in a linear form. Now, the first thing that's going to happen in glycolysis is we're going to have to invest a little bit of energy. So this very first part of glycolysis is called the energy investment phase. And the reason is this is going to cost us a little bit of cellular money in the form of ATP. So I'm going to write the word cost right here. We're going to take 2 ATP, which stands for adenosine triphosphate, and we're going to peel off a phosphate of each of those, and so that at the end we'll have 2 ADP, which would be adenosine diphosphate. So here's what happens. On these, there we go, on these 2 ATP, we're going to take 1 phosphate group off of each of them, and we're going to tack it on to the end of the glucose molecule so that you're going to end up with something that looks like this. Hopefully you can see that we have taken a phosphate off of each of these ATP and we've stuck it on the end of these molecules here. Now what that does is it destabilizes this molecule so that these will split into 2 individual molecules that look like this. Now, as we go through this process, as we go from this glucose molecule here to this molecule here, remember that all of these chemical reactions are being catalyzed by enzymes. So I'm not going to have you learn the name of these enzymes, but just remember that all of these chemical reactions that I'm showing you are being carried out by the action of enzymes, which we studied in chapter 8. Okay, so we have an enzyme that takes glucose into this form using the help of ATP. This is a bit of a cost to us. So what happens next? Well, next, we can enter into the phase where we start getting some energy back out. So this next phase is called the energy harvesting phase. The first thing that's going to happen is we are going to take one of those molecules that I mentioned in our previous lecture, NAD, and we're going to add a hydrogen and take it to NADH. Okay, this is one of our electron carriers that I've mentioned previously that is a byproduct of this process, okay? At the same time that this is happening, the enzyme that is doing this job for us is also taking a phosphate group and it's going to add it to this molecule here so that we're going to end up with our little 3-carbon sugar, and it's going to have a phosphate on each end, okay? Let's do that again over here. So there's an enzymatic reaction that occurs that, first of all, involves NAD going to NADH. Remember, this is a high energy electron carrier. This is a high energy electron carrier. So if you were paying attention in my last lecture, these are those byproducts that I mentioned that are going to be important later. This enzymatic reaction is also going to take a phosphate group, okay, and add it to the other end of this molecule so that we end up with a phosphate, 3 carbons, and a phosphate, okay? The next thing that's going to happen is, through the magic of another enzyme, we can peel each of these phosphate groups off. So what we can do is we can take 2 ADP molecules, 2 molecules of adenosine diphosphate, and we can add a phosphate group to one of them and a phosphate group to the other, and we can therefore generate 2 ATP molecules. So when we do that, when we take this phosphate off and this phosphate off, we're left with just a plain old 3-carbon molecule called pyruvate. Okay, let's do that again. So we're going to peel off each of these phosphate groups and we're going to add them each to a molecule of adenosine diphosphate, right? So this is 2. We're going to add a phosphate. So then we're able to generate 2 molecules of ATP. And by peeling these off, we're left with a simple 3-carbon sugar called pyruvate. So let's think about what we generated during this whole process. Let's think about what the products of glycolysis look like. All right, first off, we got these molecules here, didn't we? We ended up with 2 pyruvate molecules, and we're going to use that in the very next step of this process. So they're important. 2 pyruvate molecules. We also got 2 of these high energy electron carriers, NADH, okay? And then the last thing that we gained was some ATP. We made ATP here, and we made ATP here. So that's a total of 4 ATP, right? But remember that we had to invest some ATP back here at the very beginning to get this whole party started. So we used 2, but we gained 4. So our net gain from this process is 2 ATP molecules. So this is what you get out of glycolysis, okay? Remember that glycolysis, the word, literally means breaking sugar. And that's exactly what we've done. We've taken glucose, and at the very end of this process, we will have broken this into 2 3-carbon sugars. And along the way, we generated these byproducts and these byproducts. All right? Let's look at this in a summary diagram. So this is just looking at it a little bit different way. Here is glucose in its ring form, and through glycolysis, you end up with 2 ATP, 2 NADH, and 2 molecules of pyruvate, just like we talked about, okay? And if we go up here to this slide from your last lecture, here's where we are on our roadmap. We've gone all the way through here, and we have these 2 pyruvate molecules that each have 3 carbons on them. So what's going to happen next is that pyruvate is going to enter the mitochondria. All of this was in the cytosol, okay? So the pyruvate generated by glycolysis is going to enter the mitochondria. So this purple right here represents the mitochondria. Now, we've gone through glycolysis. Let's next talk about pyruvate processing. So pyruvate, I mentioned, is a 3-carbon beast. And what happens in pyruvate oxidation is you go from the 3-carbon pyruvate to a 2-carbon molecule that is attached to a very large organic molecule called coenzyme A, or CoA. So this molecule is called acetyl CoA. Now what happens during this process is, again, we're going to take an NAD molecule as part of this enzymatic reaction to get from here to here. We're going to take an NAD molecule and we're going to turn it into NADH. So we've generated another high energy electron carrier. But hopefully you notice that this process has also caused us to go from 3 carbons from our glucose down to 2 carbons. So we are also, through this enzymatic reaction, we're going to lose a molecule of carbon dioxide. So we're going to peel off one of these carbons, and it's going to combine with oxygen to turn into carbon dioxide. And this gets exhaled by you. Okay, this is a waste product. And you're just going to breathe that out of your lungs. Now remember that for every molecule of glucose, we have 2 molecules of pyruvate. So this process happens 2 times, right? This one and then one more time. So let's think about the products that are associated with pyruvate oxidation, okay? The first thing that we're going to get out of this is, let's just start down here. The first thing we're going to get out of this are 2 molecules of acetyl-CoA. We're going to use these in the next step of the process. Okay. The next thing that we get are 2 molecules of CO2. And remember, I'm saying 2 because we're going through this process twice. So 2 molecules of CO2. And the last important thing that we gain from this reaction are 2 high energy electron carriers. You know it's an electron carrier because we've added a hydrogen. Hydrogens have electrons with them, okay? So this is another high energy electron carrier. Here is that process summarized for you. So here is what pyruvate looks like in real life in terms of its chemical structure. And just like I told you, to get from pyruvate to acetyl-CoA, what you're going to do is you're going to lose a carbon dioxide. You're going to convert an NAD to an NADH. and then you'll add a coenzyme A on here, okay? And as you can see, we have 2 of these bad cats because glucose generates 2 of these for us, all right? Next step. The next step is known by many names. So in high school, you may have learned the Krebs cycle, or you may have learned the citric acid cycle, or you may have learned the tricarboxylic acid cycle. All of those are synonyms for the same biochemical synonyms for the same biochemical set of reactions. Now, some folks in some classes will ask you to memorize all of the intermediates and all of the enzymes that are involved in this process. It's pretty involved. So what I like for my students to do is to just understand what's happening to the carbons from our glucose molecule that we're following through this process. So let's start out with our acetyl-CoA. So remember, acetyl-CoA has got 2 carbons from our glucose, and it is attached to a coenzyme A molecule, which is quite big. So I'm just going to draw it like that. Now, an enzymatic reaction is going to take this 2-carbon acetyl-CoA, and it's going to combine it with another 4-carbon molecule. So there's an enzymatic reaction that's going to occur here. And it's going to combine these carbons together into a 6-carbon molecule. This particular 6-carbon molecule is a little different than glucose. It is a molecule called citrate, okay? Because I'm only drawing the carbons for you. So all of the other atoms that are involved in this are different than the ones involved in glucose. So what's happened is we've taken our 2-carbon from our acetyl-CoA and our 4 carbons here, and we're going to combine these together. This is just hanging around in the mitochondria, okay? This is not part of the glucose. This is part of the glucose. We're going to combine these together to form a citrate molecule. And what's going to happen is that this is a biochemical cycle where this 6-carbon citrate is going to be manipulated through a series of enzymatic reactions to end back up at this 4-carbon molecule. And I was going to give you the name of the 4-carbon molecule. I don't even remember it off the top of my head. It is called, hang on. Oh, yeah, it's called oxaloacetate, okay? Just in case you're looking for a name to help you understand what this molecule is, this is called oxaloacetate. Okay, I will not test you on that, but if that helps you to understand this, then great. So the 2 carbons from your acetyl-CoA molecule combine with the 4 carbons from an oxaloacetate molecule via an enzymatic reaction to form the 6-carbon citrate here. Now, this series of biochemical reactions to get us back to here are going to cause us to have a few byproducts made. So the first thing that's going to happen, you're going to lose 2 CO2. Well, look at this. We only had 2 carbons here in the first place, didn't we? So that's interesting. Throughout this process, you're going to take 3 NAD molecules and you're going to convert them into 3 NADH molecules. So you're going to gain some high energy electron carriers. There's another type of high energy electron carrier you're going to gain throughout this process. You're going to take a molecule called FAD, and it's going to get converted to something called FADH, which has actually two H's there. You're going to get one of those. And the very last thing that you're going to get out of this is you're going to go from an ADP to an ATP. So what I'm telling you is that the process of going from citrate going through all these biochemical reactions to get back around here and regenerate this 4-carbon molecule to start this process again, results in all of these byproducts, okay? So let's look at the products of the citric acid cycle. Okay, so I said that you get 2 carbon dioxide for an acetyl-CoA. But again, remember that for every molecule of glucose, this cycle is going to happen 2 times because we had 2 molecules of acetyl-CoA to go into this, didn't we? So you're going to end up with 4 CO2 being lost. That's interesting. Why is that interesting? Let's go back a second. When we talked about pyruvate processing, we lost 2 carbons in CO2 here. Now, in the citric acid cycle, we've lost 4 more carbons. Remember that our glucose molecule only started out with 6 carbons. So at this point, we have lost or digested or oxidized, however you want to think about this. We have lost all of our glucose. We have used the glucose up. Okay, and you're going to exhale this out. This citric acid cycle also results in 3 NADH for each turn. So remember, you've got 2 turns, so you're going to get 6 high energy electron carriers in the form of NADH. You're also going to get 2 FADH pluses, aren't you? Or sorry, FADH2s, because again, this turns 2 times. And the last thing that you're going to get is you're going to get 2 molecules of ATP. Because again, this thing is turning 2 times. So for every molecule of glucose that goes into this system, these are the products that the citric acid cycle generates. Now, I think it's helpful to look back up a bit and look at an overview, okay? So this is an image from your book, and let's look at what's going on here. So here we have our glucose molecule. It is 6 carbons, right? 1, 2, 3, 4, 5, 6. Glycolysis is going to break it into 2 3-carbon molecules. And the process of going from 6 carbons to 2 3-carbons is going to generate some byproducts for us. It's going to generate 2 high-energy electron carriers in the form of NADH, and it's going to generate 2 ATP for us. All right? Then these 3 carbon molecules, these pyruvates, are going to get shuttled into the mitochondria, okay? Once they get into the mitochondria, another chemical reaction is going to occur, another enzymatic reaction. And this enzymatic reaction is going to result in our 3-carbon pyruvates being converted into 2-carbon acetyl-CoA molecules, like I drew for you before, okay? So you can see that going from 3 carbons to 2 carbons means we've lost some. So we lost a carbon on this one, we lost a carbon right there. It gets breathed out in the form of carbon dioxide. You exhale this, right? This is a waste product. This enzymatic reaction also generates for us 2 more of those high energy electron carriers in the form of NADH. Now, what's going to happen next is that each of these acetyl-CoA molecules is going to feed into this citric acid cycle. And for each turn of the citric acid cycle, we get 6 NADHs, 2 FADH2s, so these are high energy electron carriers. We lose 4 carbons, 1, 2, 3, 4. We're going to lose them in the form of CO2, which again is waste. You exhale this. And we're going to gain 2 ATP. So a few things to point out. First of all, at this point, we have oxidized the entire glucose molecule. We've used up all of the carbon bonds here that we're storing energy. So we've gone from 6 carbons in the glucose molecule to 6 CO2s here and here. So glucose, at this point, glucose is completely oxidized. It's all gone. So from this point on, the story is going to be all about these high energy electron carriers. These guys are what we're going to talk about next. The next thing that I want to point out to you is that up to this point, we've made relatively little ATP. We've only made a total of 2 here and 2 here. So 4 ATP so far. That's really not very much. So what I want you to know where we're going is that we're going to use these high energy electron carriers here. We're going to put them in the electron transport chain, and we're going to use them to make a lot more ATP. And I'm going to show you how that happens. But before I do, I want to take a step back and I want to think about where all of this is happening. So let's take a look at the mitochondria. You folks know that the mitochondria is, of course, the powerhouse of the cell. So it is where ATP is being made. So let's write that down. Mitochondria is where ATP is being made. So I'm going to put ATP synthesis. Right? Which is why we call it the powerhouse of the cell. Now let's look at it up close. So first of all, I'm going to draw you like a cell just to kind of get you oriented. So this is just a general like animal cell. And over here, I'm going to draw you a nucleus, and I'll put an N in it so you know that's the nucleus. Now I'm going to draw a giant mitochondria for you. All right. Mitochondria in the cell are not this big, right? They're much smaller than this, but I'm enlarging it to show you some features. The first feature that I want you to notice is, of course, that it is double membraned and that these internal membranes are highly folded. So if we're looking at these double membranes, please remember that this membrane here is a phospholipid bilayer. So if I were to actually draw this out, I would have our little phospholipids, right? So all of this is one phospholipid bilayer, and all of this here is a second phospholipid bilayer. So we have a system of 2 membranes, okay? This area here is called the inner membrane space, okay? And so I'm just going to put down here so you guys can write that out. The inner, or sorry, not inner, excuse me. The intermembrane space. Okay. So the space between this inside membrane and this outside membrane is the inter, for in between, membrane space. The interior here is called the matrix. And then the outer membrane is just called the outer membrane. The inner membrane is just called the inner membrane. Now, up to this point, in thinking about cellular respiration, our glycolysis happens out here in the cytosol. Glycolysis. But The pyruvate oxidations, I'm going to put PO for pyruvate oxidation. That's happening in the matrix of the mitochondria. And the citric acid cycle is also happening in the matrix of the mitochondria. So the next place that we're going is our fourth step of cellular respiration called the electron transport chain. Where is that occurring? Well, the enzymes and the proteins involved in the electron transport chain are actually embedded in this inner membrane here, okay? So all the proteins for what we're about to talk about next occur on this inside membrane. And that is why we see that the membranes of mitochondria are so highly folded, okay? So the reason that the inner membrane is highly folded is to increase surface area. Now, what do I mean by that? Well, let's think about this. If I fold up and just pack as much membrane as I can in this mitochondria, then I literally have more space to park my electron transport chain proteins. And I can just put tons and tons of them on here so that we can generate all kinds of ATP in your cells, okay? So this is actually, this folding of the inner membrane is an adaptation. And it's an adaptation. And it's an adaptation to increase the rate of ATP synthesis, okay? Because the more membrane we have, the more space we have to park these electron transport chain proteins, and the more ATP we can make. Now, incidentally, just so you know, this is one version of a mitochondria from your book, and all that this is a different drawing, okay? I don't want to confuse anyone. This is just sort of highlighting for you what this highly folded membrane looks like, okay? And that you have an outer membrane and an inner membrane space, okay? So let's focus in on what's happening in the electron transport chain. So let me first draw us a diagram. We're going to draw one membrane, okay? Oops. And in my example, this is going to be, this right here is going to be the outer membrane of a mitochondria, okay? So that means out here is the cytosol. Now we're going to draw an inner membrane, and I'm going to leave some spaces because we're going to insert some things in these spaces. Okay, but this is supposed to be the inner membrane. Okay, this then becomes the intermembrane space. And this becomes the mitochondrial matrix. So again, just to review, glycolysis is happening out here. When glycolysis makes pyruvate, it gets shuttled in here. Oops. Pyruvate. So that's where pyruvate oxidation occurs. This is also where the citric acid cycle occurs, okay? So the next thing that's going to happen is the electron transport chain. Now remember that we have all those high energy electron carriers that we generated through our previous biochemical reactions. So what's going to happen is, let's take one of these high energy electron carriers. Let's take an NADH. And this protein here that's embedded inside this inner membrane is going to peel off an electron, okay, so that this goes from being NADH and becomes NAD all over again. That's actually kind of important. Now we've regenerated some NAD that can go back into our earlier reactions, like our pyruvate oxidation or glycolysis or the citric acid cycle. So we're actually regenerating some of the things that we need to keep these processes going. All right, so what we've done so far is we've taken an electron off of here. And in doing that, this electron has some energy associated with it, and it can pump this hydrogen that was on NADH, this proton, it can pump it from the matrix to the intermembrane space. So that's pretty cool. Now, what's going to happen next is this electron has some energy associated with it. So we're going to use it again. We're going to actually transport it from here over here to a new protein. I'm actually going to change colors so that y'all can see that. So we're going to transport this electron from here over this direction to a whole new protein. And this transportation is done by a special molecule called Q. Q, you can think of as sort of a molecular shuttle bus. So it's a shuttle, and it moves electrons around for us. Q is an abbreviation. The real name for Q is ubiquinone. Okay. So Q, or ubiquinone, picks up this electron and is going to transport it over to another protein in the membrane. So here it is. Here's our electron again. Now, this electron has some energy with it. So what's it going to do? It's going to pump a hydrogen, just like we did over here. It's going to pump a proton, a hydrogen ion, up into this intermembrane space, okay? Now, what we're doing in both of these circumstances is we're using the energy of these electrons to pump against a concentration gradient. So we're going to make a whole bunch of protons up in this area. We're going to make a really high concentration up here, and we're going to have a low concentration down here. All right. Now, this electron still has a little bit of energy left in it. We've used up some energy. We had a bunch of energy in this electron, brought it over here, had some energy in this electron. We're going to actually move it one more time using Q again. We're going to move it over here. So again, this is done by that molecular shuttle called Q. So this electron now has just a wee bit of energy left, and we have just enough energy to do one more pumping action so that we can get one more proton up into this intermembrane space. But at this point, we have exhausted our poor little electron. It's done. We can't use it anymore for any more hydrogen pumping. So now we've got to put it somewhere. This electron has to go somewhere. It can't just be sitting out here by itself. Where this electron goes is a molecule of oxygen. Okay, that is an O for oxygen. So this electron is going to be dumped onto an oxygen. This oxygen is then going to combine with two other hydrogens that are in the mitochondrial matrix to form something called metabolic water. So you actually make a little bit of water in your mitochondria through this process where we dump an electron onto oxygen. This oxygen then combines with a couple of hydrogen to make this water. Now, this isn't enough water that you can live on it, okay? So I'm not telling you that you don't need to drink your water and hydrate. I'm simply telling you that you make a little bit during this process. Now, this feature here, this is important to me, okay? So I will almost certainly ask you about this on an exam. I like to think of this as a molecular dumping ground. The big thing that you need to remember, and you need to write this down, is that oxygen, oxygen is the final electron receptor in the electron transport chain, okay? So where did this oxygen come from? Well, it came from the air. This is why you breathe, okay? So let's put why you breathe. This is why you suck in oxygen on a regular basis, okay? So this is really important. This is why you take in oxygen. You take in oxygen so that you have a place for these electrons to go. You breathe out carbon dioxide because you're getting rid of the waste products of glucose oxidation. So your entire respiratory system has pretty much been explained to you now, or at least why you take in the things you take in and why you breathe out the things you breathe out, okay? Now notice I've got one more little space here. So let's draw in one more membrane protein. And this one's going to function just the same way, only instead of accepting an NADH high energy electron carrier, this one accepts FADH2. Remember that guy? We generated two of those in the citric acid cycle. And so this protein strips, that's an FAD, FAD. This is going to strip an electron off of that molecule, and it's going to use the energy of that electron to pump hydrogen ions in this intermembrane space. Then just like before, we're going to shuttle this electron over to the next protein, where once again, it still has a little bit of energy, and we can use it to pump some hydrogen ions into this space. And then again, we'll do another shuttle. Do it in red again. Another shuttle, boom. Move that electron over here. Use it once again to get some pumping done. And then when it's all finished, this guy is exhausted and depleted of energy. He'll get dumped onto an oxygen molecule, which will combine with hydrogen and make some metabolic water. Now, so far, what we have accomplished is we've used the energy in our high energy electron carriers to build up a ginormous hydrogen ion gradient. We haven't made any ATP yet, but what we have done is made a really strong hydrogen ion gradient. This intermembrane space is going to end up being pumped full of these hydrogen ions based off the energy of our electron carriers, okay? So let's look at that in a little bit cleaner version. This is a figure from your book. So you can see we have our high energy electron carrier. We're peeling off an electron. The electron is getting, first of all, the electron's energy is pumping hydrogen ions into this intermembrane space. Then we're going to shuttle this electron using this Q molecule, shuttle, shuttle, shuttle over here. We're going to use its energy to do some more pumping. We're going to move it over one more time, use some energy to do some more pumping. And then eventually these electrons are going to be exhausted and they're going to get dumped onto an oxygen, which will combine with some hydrogens to form water, okay? Here's the same thing with FADH2. We're going to peel off an electron. We're going to use Q to shuttle it. All the while, we're pumping hydrogens, and again, dumping the electrons onto an oxygen and forming water. Up until this point, we have not made any ATP yet. So that's what we're going to do next. So if I were to continue on this, like draw this out here, just extend this membrane, there would be another protein over here, okay? And that is this protein right here. So over here are all of the other electron transport chain proteins that we've been looking at. But also embedded in the membrane is this guy right here, and his name is ATP synthase. Incidentally, anytime you have a molecule with the suffix ASE on it, that tells you that that molecule is an enzyme. So any molecule that ends in that suffix is an enzyme. ATP synthase is an enzyme that synthesizes ATP, okay? So how does it work? Well, we have a really high concentration of protons up here, and at this point, we have a low concentration of protons here. So we know from our studies of diffusion that things like to go from areas of high concentration to areas of low concentration. So what's going to happen is that these hydrogens are going to move through this ATP synthase molecule, and it's actually divided into two parts. This unit here, which is where the hydrogens can enter, it's kind of like a channel protein, and this unit here. And what happens is as these hydrogens diffuse back through here, they create an action. Their positive charges and they're flowing through here actually creates almost like an electrical current, kind of, sort of. It's sort of like how a dam creates electrical current. You know, if you have water flowing over a turbine, basically what happens is these flow through here, and as they do, they cause this thing to spin around and around and around like a motor. This is actually a little molecular rotator shaft. So this thing spins like crazy. If you could see this under microscopy, you can actually watch this twirling. And the action of this twirling is actually used to drive a chemical reaction where we take adenosine diphosphate, so 2 phosphate groups, then we take an extra phosphate group, add these together, and we can form ATP. So we're going to use this hydrogen ion gradient to form ATP. So let's write that down. So use the hydrogen ion gradient The hydrogen ions are going to go from an area of high concentration to low concentration. As they are moving from high to low concentration, they move through this ATP synthase. I'm going to abbreviate. They move through ATP synthase. As they move through, they cause it to spin. And the spinning action takes ADP plus a phosphate group and turns it into ATP. So at this point, we have achieved our goal. The whole goal of cellular respiration was to start out with a glucose molecule, right? And there's some energy stored in the bonds of glucose, but we can't use that in our cell. What we use in our cell for energy is ATP. So the goal of cellular respiration was to take a glucose molecule and turn it into ATP. And at this point, we have done that. Now the question becomes, how much ATP can we make? How much can we make using this process? Well, let's take a look. It turns out, let's see here, is this where I wanted to, yeah, this is where I wanted to do this. Okay, so it turns out that we said that we had some high energy electron carriers. We have 2 NADH that we got from glycolysis. We have 2 NADH that we got from pyruvate oxidation, and we have 6 NADH that we got from the citric acid cycle. And if we use these guys in the electron transport chain, it turns out that for every NADH molecule that we put into the electron transport chain, you can make 3 ATP. So if we are oxidizing glucose and we add all these up, 6, 7, 8, 9, 10. For every molecule of glucose, we have 10 NADHs, 10 times 3, that's 30 molecules of ATP that we can make using the NADH byproducts that we get from breaking down glucose. Let's look at this guy here, this FADH2. It turns out that if we use FADH2 and we put it into the electron transport chain, you can make about 2 ATP for every FADH2 that you put into this right here, okay? Now, we got 2 FADH from our citric acid cycle. So 2 times 2 means that using these high energy electron carriers from 1 molecule of glucose, we get 4 additional ATP. So these are the ATP that we get from our high energy electron carriers, but let's not forget that we generated just a little bit of ATP from glycolysis and from the citric acid cycle. So we had 4 ATP that we already made, right? So from a single molecule of glucose, we can end up with 38 ATP. Wowza. That's a lot of ATP from a single molecule of glucose. Now, this is a theoretical number, okay? This is how much in theory we should get. But researchers can actually measure how much ATP is generated from this process. And it turns out that if you look at how much is made, on average, we only end up getting about 29 ATP from this process. But by theory, we should be getting 38. So where are some of these ATP going? Well, remember that we are actually moving some stuff into the mitochondria. We're moving some NADH into here. We're moving our pyruvate into here. And it turns out that moving stuff into the mitochondria costs a little ATP. Costs ATP to shuttle molecules into the mitochondria. Oops, well, sheesh, molecules into mitochondria. Okay, so in theory, we get this much. Turns out we usually only generate this much, but that's still a lot of ATP that we get out of this process. Now, the reason I want to highlight how much ATP we get out of this process is I want to switch gears here at the very end of this lecture. So cellular respiration, I just told you, in theory, generates 38 ATP. In real life, we often get somewhere around 29 ATP out of this process. And remember that cellular respiration is an aerobic respiration, meaning that it requires oxygen, right? And you guys saw that oxygen's role in this is that oxygen is the final electron acceptor in the electron transport chain. But what if we are in a situation where we don't have any oxygen? Can we still turn glucose into ATP? If there's no oxygen, can you still take glucose, and trade it in to make some ATP? Can you still do that? The answer is yes, you can, but you make a lot less ATP, okay? As a matter of fact, you only get 2 ATP per glucose if you have no oxygen, compared to 38 ATP per glucose if you have oxygen, okay? So this is anaerobic or without oxygen. Now let's look at why that is, okay? Without oxygen, in anaerobic respiration, what happens is you can still do the first part of cellular respiration. Remember that the first part of cellular respiration is glycolysis. Okay, so you can still do glycolysis, which is the first part of cellular respiration. And if you remember, in glycolysis, you generate 2 ATP. You also generate 2 NADH, and you generate 2 pyruvate. This is review. We've already covered this. So this part can happen even when there's no oxygen. Glycolysis can happen without oxygen. So in a pinch, if you're low on oxygen, you can just do glycolysis. You're only going to get 2 ATP out of it, but you can still do something. However, we are left with a problem. And here's our problem. If we just keep doing glycolysis, right, and we never go through the whole process all the way through to the electron transport chain, we keep making NADH, but we never regenerate the NAD. Let me go back for just a second. Here, we took NADH, we stripped off an electron, and we took it down to NAD. This NAD can be fed back into glycolysis to keep this whole process going, right? It can be fed back into pyruvate oxidation. It can be fed back into the citric acid cycle. So you're regenerating this stuff here to feed back into the first 3 phases of cellular respiration. But in this example here, oops, sorry, wrong slide. In this example here, we're running glycolysis. We're never getting to the electron transport chain because we don't have oxygen. So we're using up our NAD and we're taking it to NADH, and NADH is building up and building up and building up because it's not going to the electron transport chain. So we have a problem. To keep this process going, we need to have some way to take these NADHs and turn them back into NADs so that we can have glycolysis go, make some ATP, turn this back into NAD, have glycolysis go, make 2 ATP, turn this back into, you see what I'm doing here? So here's how we manage this. We manage it through a process called fermentation. Okay, what happens in fermentation is there is an enzyme, an enzymatic reaction that converts pyruvate into a molecule called lactate or lactic acid. Okay, so there's an enzyme, it's actually called lactate dehydrogenase. And it converts pyruvate here into lactic acid here, okay? Now, in doing that, what it does is it regenerates this NAD here. So now we can have NAD go through glycolysis, make some pyruvate, turn it into lactic acid, which regenerates this. We can go back. Lactic acid regenerates this, go back. But what you end up with is you do end up with a buildup of lactic acid. This is why your muscles get sore when you exercise real hard. So if you're exercising really strenuously and you're like just running or lifting weights or whatever, and you start becoming short of breath. Well, the reason you're short of breath, the reason that you're breathing heavily is because you need oxygen so that you can make ATP. But sometimes you can't keep up. Sometimes you need more oxygen than you can breathe in. So you start converting to anaerobic respiration, okay? When you convert to anaerobic respiration, you build up this lactic acid in your muscles, and this lactic acid makes your muscles sore. This is why your muscles get sore, because you've been doing a little bit of anaerobic respiration when you're exercising strenuously, okay? Now let's see here, what do I want to talk about? This slide. Okay, there's different kinds of fermentation. Yeah, I had a note here from a previous lecture. There are different types of fermentation. The one that I described to you is the one that happens in your muscles. But here's an example of fermentation that takes glycolysis and it takes pyruvate and it converts pyruvate into ethanol, which is the alcohol we drink. Okay, so this process goes through and it regenerates our NAD so we can keep glycolysis going, okay? But the end product of this type of fermentation is alcohol. This is done by yeast and bacteria. So you can have some bacteria that do this. You can have yeast that do this. So like if you're making wine or beer, that's how this process is done, through yeast that are doing anaerobic bacteria. This is how you make wine and how you make beer. The difference between this fermentation and the one that happens in your muscles is simply the part that regenerates the NAD+. So you can see that in this version, we're taking pyruvate and we're going through a different series of enzymatic reactions to get a very different end product, ethanol. Here, we have an enzymatic reaction that gives us lactate. But the general concept is the same. In both examples, we are regenerating our NAD+ to keep glycolysis going so that we can keep up our anaerobic respiration. But at the end of the day, the thing that you need to remember is that aerobic respiration gives you, I'm going to put 38 because that's our theoretical yield, 38 ATP per glucose. Anaerobic or fermentation gives you 2 ATP per glucose. That is why larger organisms that are very active, most of the organisms that you'll study in biology, most of them are what we call aerobes, meaning that they need oxygen in order to maintain their energy, okay? This is why aerobic respiration is so common. Now, anaerobic respiration does still occur, like I told you, if you're exercising strenuously, and it can occur in yeast. It occurs a lot in bacteria. So little unicellular beasties do just fine using anaerobic respiration because they're smaller. And in that way, bacteria are able to live in some places where there's no oxygen. So for example, if you have a bacterial infection like under your skin and you get a cyst or something like that, those bacteria are living in an environment without oxygen, but they're doing just fine, okay? Because they're using fermentation that we just looked at in order to get their ATP. Now, I did want to mention a couple of different vocabulary terms. If you are an anaerobic organism that that only does anaerobic respiration, you're called an obligate anaerobe. If you're one that can sometimes do aerobic and sometimes do anaerobic, depending on what the situation requires, you are called a facultative anaerobe. Similarly, you can be an obligate aerobe, meaning you have to have oxygen in order to get sufficient energy. I would say that humans are obligate aerobes. You can do a little bit of anaerobic respiration, but not for long. You would die pretty quick in the absence of oxygen, right? So this really applies more to bacteria. All right, so this is the end of what you need to know for Chapter 9. Happy studying for your quiz, and I'll talk to you next time. Bye-bye.